Is it possible to regrow neurons after they've been severely damaged or even crushed? Scientists may have found an approach that allows them to regrow a certain type of neuron called retinal ganglion cells. These neurons transmit information between the eye and the brain. Stay tuned for some more cool science.
Cool science.
I mentioned the awesome possibility of regrowing RGCs. But how do RGCs grow to become RGCs in the first place? If you go back in time, the life of a cell, all the way back to embryonic development, you see that all cells look the same. That's because they all share the same instruction manual, the same DNA. So how do these identical cells end up being so different from each other, becoming muscle cells or skin cells or heart cells? At a certain point, cells begin to read only a small specific portion of their genes, which allows them to synthesize certain proteins. The proteins that they synthesize determine the identity of the cells. So for example, cells that are developing into RGCs would only read the portion of their genome that pertains to all the proteins necessary to become an RGC. Wait, but how do RGCs avoid reading the wrong genes that would turn them into, say, skin cells or heart cells? The cell uses a special genetic roadblock called methyl. Methyl attaches to all the genes that are unnecessary and that don't need to be read. And when the cell goes through the genetic sequence, it skips over those methylated genes. When the cell ages, however, it starts making mistakes and putting up methyl roadblocks on important necessary genes. Since those roadblocks are in place, the cell cannot read the genes that it needs to read, and therefore it can't synthesize the proteins that it needs to properly function. That's why scientists refer to methylation as a marker for aging. So scientists thought, if we could find a way to take off these methyl roadblocks that were put up by mistake, then we could bring the cell back to a youthful state. Or in other words, we could reverse cellular aging. In this youthful state, there would be rapid growth, which would allow damaged neurons to be regenerated. And scientists found a combination of proteins called OSK transcription factors that can take down these mistaken methyl roadblocks. Researchers took mice with damaged RGCs, and when they injected them with this protein OSK, they saw that the cells were able to regenerate themselves. And here's the amazing part. Researchers found that OSK can induce long-distance regeneration of RGCs, and it could do it in both young and old mice. It could do it before and after injury, and it could also do it in disease models like glaucoma. Most importantly, the injection of OSK didn't lead to any unwanted changes in cellular functioning, which is a very important point since we're dealing here with an epigenetic manipulation. This is a remarkable breakthrough for the field of epigenetics, and it would be fascinating to continue exploring the effects of OSK and answer new questions like, how does OSK selectively remove methylation? Can OSK have similar effects on different types of neurons? And can OSK have these miraculous effects on in vivo human neurons? Thank you for joining me today, and I'll see you next time.